The next process is translation. After transcription, we know the translation. Translation means from mRNA, the protein will now synthesis. We know the central dogma. First DNA to RNA. First DNA to DNA, that is called replication. From DNA to mRNA, that is called transcription. From mRNA, protein will synthesis. And this process is called the translation. That is, we will learn now. Okay, so first translation, the activation of amino acid. So for the activation of amino acid, what we will require? We know a protein is made up of many amino acid. A protein is made up of many amino acids. So amino acids are monomer of a protein, right? So for protein synthesis, amino acid is required. For activation of this amino acid, ATP is required, and enzyme is required, that is called amino acyl synthetase. Okay, so this amino acid first bind with this ATP and this with this enzyme, and they form a activated amino acid, that is called amino acyl. So what I am telling you, for this activation of amino acid, ATP is required and an enzyme is required, that is amino acyl synthetase. So first the amino acid, any amino acid that is in inactive form, they take ATP and this enzyme and they form an activated amino acid. This activated amino acid is called amino acyl. So this is in the form AMP with the enzyme. This is adenosine triphosphate, there adenosine monophosphate. So 2 phosphate they lose. That's why we have written this with plus 2 phosphate. Okay. So now what happened? The active, or as I told you, AUG, it's a start codon, right? It code for methionine amino acid. So the first amino acid, that is methionine, it is activated amino is always in the form of activated because it is called the start codon. Okay, when we will see the protein synthesis start, these methionine amino acid will synthesis fast, and its codes for particular, its codon is AUG. So AUG, this codon is responsible for synthesis of methionine. This is always in the active form, and this is called F-mate. Okay, this is called F-mate. This is the first amino acid which will synthesis when protein synthesis will begin. So this is first, in the first step, the activation of amino acid done. Next, the transfer of activated amino acids to tRNA. Okay, so we know already we have learned the structure of tRNA here. The structure of tRNA here, we have learned that this is a 3' end, this is a 5' end. Here the amino acid binding site, this is T loop, their ribosome recognition side, and this is the anticodon side, and this is the D loop. So we have already learned the structure, right? So here, so first this amino acid will bind with tRNA, right? So where is tRNA amino acid will bind? We know in the 3' end there is a OH group. This is the amino acid. This is the amino acid binding site. So first here the amino acid will bind. Okay, so you see, so amino acid binds with tRNA, and then this AMP, the enzyme, all will get separated. Okay, they are not in together, they are now separated. So amino acid with tRNA will bind fast. So the role of tRNA is transfer of amino acid to the site of protein synthesis. So what is the role of tRNA? Is a transfer of amino acid to the site of protein synthesis. Okay, so now the main step, that is the protein synthesis, will begin. So for this protein synthesis, what we will done? The assembly of ribosomal subunits. Assembly of ribosomal subunit, we already know that ribosome have two subunit. One is the smaller subunit, one is the larger subunit. In smaller subunit, what's happened? The mRNA binding site. In the smaller subunit, mRNA will bind. In the larger subunit, there are 3 sites: A, E, P. Also, we can say EPA. E, the exit site. P is for the peptidal site, means peptide bond will be formed here, and E is the exit site, A is the amino acyl site. Amino acyl means the enzyme will bind here. Okay, so these two subunits, when these two subunits bind, when protein synthesis will begin. We have already learned this part, structure of ribosome in the chapter cell in class 11 biology. So these two subunits bind when protein synthesis will begin. So assembly of ribosome will take place when protein synthesis will begin. Assembly means these two smaller subunit will bind. And here is another very important thing, that is magnesium ion is required, and it is required in a particular amount. Less amount of magnesium or more amount of magnesium will not help in binding of these two subunits. Okay, so a particular amount of magnesium ion is required for binding these two subunits, and when the protein synthesis, when these two ribosomal subunits will bind, when protein synthesis will begin. So now it is the first step. And the next step, what happened? The next step, the attachment of mRNA. So now the mRNA, as I told you, the mRNA will bind in the smaller subunit. So the mRNA will bind in the smaller subunit. In the third step, the polypeptide synthesis or the protein synthesis. The polypeptide or protein will now synthesis here. So this is the smaller subunit, this is the larger subunit. Suppose this is mRNA, the codon for we have already learned the structure of mRNA, right, in the previous class. So suppose this is the codon part. We have already learned the structure of mRNA. In mRNA, there is a start codon, there is a stop codon. So start codon means the AUG, the methionine amino acid will start here, and the stop codon, we have already learned the 3 stop codon, right? They will not code for any protein synthesis, right? So, and here the middle part, the codons are present. The codons are present. We will responsible for synthesis of a particular code, right? So here first, as I written here, you see the first this is the first, this is the start codon, right? AUG, it is a start codon. As I told you, AUG is the start codon. This is called methionine, and also we call this AFMAT. Okay, acha. And suppose the next codons are UUU, CAU. This is, so the first codon, the start codon will attach at the site of P. P means the polypeptide site, polypeptidal site. Okay, so now all codes for amino acid read at the site A. So all amino acid codes will read at the site A except AUG, because we know this is the start codon. Okay, in the site A, all the codon will read here one by one. We will see the process now. Now you see the FMA containing tRNA bind at site P. So suppose this is, as I told you, in the site, this is the 3' end and this is the 5' end, and 3' end OH is the functional group. That's why anything, whatever the thing will take place at the site OH, 3' end, where OH the functional group is present. So the first amino acid will bind here. Okay, the first amino acid will bind here. So the first amino acid come at the site P. Okay, so the first amino, and we know the code is AUG. So this is the P will recognize, yes, this is the codon for FMAT, means methionine amino acids. Okay, so it will recognize this. And the next amino acid, suppose next amino acid containing tRNA again will come at the site A. Suppose this is AUG, the next code is UUU, the next code is CAU. In this way the codon will be go. Okay, and at last there will be the stop codon. Okay, acha. So the next tRNA containing next amino acid, suppose this is amino acid A, this is amino acid 2, because this is FMAT, this is an amino acid for methionine, and this is amino acid 2 containing tRNA. So these will bind at next site, that is A. Okay, so K, A, as I told you now, here is all codes for amino acid and read at the site A. At site A, this is called, the site A is also known as amino acid site. Here all the codes for amino acid will read here. So the next amino acid containing tRNA will bind here. So what will take place in the next part? Translocation. What will means ribosome move on mRNA further. Ribosome will move. So suppose here is the ribosome will move in this way. Ribosome ribosome, the smaller subunit will move in this way. So when the smaller subunit move in this way, what will happen? This first tRNA containing F-mate amino acid, it will come on the site exit site. Okay, so exit site is come. As I told you, exit site means the tRNA will now move, and at P site, that is a polypeptide site, peptidase site, so there will be with the next amino acid there will be a bond, polypeptide bond will form. So at exit site, the code AUG is also moved. First it is at site P, now it is at site AUG, site E, it moves and the bond forms. Next tRNA comes at the site P, because the mRNA is moving, so it shifts here, this shift here. So now this tRNA shifts at P part, and obviously the next codon will come at site P. So the next codon UUU come at site P. So it's recognized for the next amino, suppose UUU, whatever the amino acid it is, poly, as I told you, UUU, it's code for UUU codes for phenylalanine. So the phenylalanine amino acid will come here. Okay, so again it is make a bond. In the next part, this will come, this will move further. This tRNA will come at exit site. The tRNA part will remove. F-mate will bond with A2 means the second amino acid. Okay, so now UUU will move at exit site. Whatever this thing happen, it will goes on that part, right? From here it will shift here. The next amino acid come at P. It reads for particular amino acid, the bond formation takes place, then it will shift at exit part. In this way the process will continue and mRNA will shift. So the next amino acid, suppose that is containing tRNA amino acid 3. Suppose its code is CAU. So in the next part what will happen? You see AUG, next UUU come at exit site, like CAU come at P site, right? P site. So the bond forms. Next this will go on exit site. The next amino acid will come. In this way the process will continue until the stop codon comes. Suppose here UAG, the stop codon. Okay, so I have written only 3 amino acid, but suppose it is many amino acids are there, the process will continue and continue until the stop codon comes. We know in the A structure of mRNA, the first part is the start codon, here is the stop codon, and the middle there are many codons that are responsible for synthesis of protein. So in this way the mRNA, the codes are going on until the stop codon. This is the stop codon comes, then the protein synthesis will stop, because we know so many amino acid will bind and they will make a protein, and between this amino acid what bond is forming? Peptide bond is forming. Okay, so stop codon, suppose UAG is a stop codon. I discussed with you here UAG, UGA, UAA. These are the 3 stop codons. Suppose one stop codon UAG, this is also a stop codon, comes here, then it will not responsible for synthesis of any amino acid. So the protein synthesis will stop here. Okay, so in F-mate, the first amino acid, second amino acid, third amino acid, in this way it is continue until the stop codon comes, and when the stop codon comes, the protein synthesis will stop here. So this is the process translation. In this way protein synthesis takes place in our body.